All right, guys. So let's get started with this review for Unit 2. These are the questions that I got from a few of you guys. So, not too many people sent me questions, but there are a good number here. And yeah, I think having a review online works out better for all of us. That way, I don't have to drive over there for just a few people to benefit. This way, those who really want to benefit from a review can just log on and or send me questions and then watch the review online. Okay, so let's get started. Let's start. We're just going to go down the list and go through it. This question is from chapter 6 on cancer. How do proto-oncogenes become oncogenes? And we covered this in this slide. So just FYI, the way I'm going to do this, I'm not going to animate the slides. I'm just going to show you the slide where you can find the answer to the question or where the content that pertains to the question is found, and you guys can look through that slide and understand it. And obviously, some of these slides we covered on online lectures. Just look up what content was covered in the online lectures, and you can probably find the online lecture for that. Um, but okay, so oncogenes, we covered this in. This is one of two types of genes that are mutated in cancer. You have oncogenes and tumor suppressor genes. So oncogenes, before they become oncogenes, are called proto-oncogenes. Proto comes from before. Oncogenes, onco, the word onco means tumor, or is the same root for tumor. And then genes. So they're oncogenes are genes that cause tumors. Proto-oncogenes are before they're activated. And normally, proto-oncogenes are genes that promote growth and division. So they're normal. You want them in your body. They are the ones that kick in when your cells are to tell your cells, all right, it's time to divide. These are the genes that promote division and growth. But when they're mutated in cancer, so these genes can acquire a mutation. Again, it can be a random mutation that happens during DNA replication, or a mutation caused by a carcinogen like UV rays from the sun or cigarette smoke in your lungs. But once a proto-oncogene acquires a mutation that activates that gene, it becomes an oncogene. So now that oncogene can basically overstimulate cells to divide, and that can cause a tumor. Okay, so again, how do proto-oncogenes become oncogenes? By mutating a proto-oncogene by a mutation. All right, what enzyme is in charge of DNA replication? So now we're getting into mitosis and meiosis, and this we saw on the second part of the mitosis meiosis lectures. And there's two things that have to happen before a cell divides. One is they must copy their DNA or replicate their DNA, and two is they have to condense that DNA into chromosomes. And that's the process called DNA condensation. And at what stages do these happen? Replication happens in the S phase, synthesis phase, and condensation happens in prophase, which is the first of the four steps in mitosis. And this is the procedure or the process by which DNA is replicated. The two strands are, the DNA is unwound, the two strands are separated, and then this enzyme called DNA polymerase synthesizes new strands using the parental strands as a template. So if in the diagram on the right, the parental strands are the red ones, and those are going to be the template, what the DNA polymerase is going to use to add nucleotides to the other strand. So you can see that by the end of replication, you have now two new molecules, but only one of the strands for each molecule is from the original DNA molecule. So this is called semi-conservative replication. Only one original strand in each new chromosome is conserved from the previous DNA molecule. And again, the question was what enzyme is in charge of DNA replication, and it's this one, DNA polymerase. Okay, why is it important that meiosis produce haploid cells? So we saw this is a slide actually from lecture 11, but it illustrates the point. So here, the male and the female have generated their gametes via meiosis. So their egg and the sperm, they're going to be haploid. They're going to have half the normal content, so that when they fertilize, they can form a diploid organism, a zygote that has 46 chromosomes. So it's important to create haploid gametes so that when they come together in fertilization, they form a diploid organism that now has the full 2 copies of each chromosome. All right, what is the difference between metaphase and metaphase 1? So here you need to realize that metaphase is a step in mitosis, whereas metaphase 1 is a step in meiosis, right? Remember, meiosis goes through 2 rounds of cell division, meiosis 1 and meiosis 2. And so metaphase 1 is the metaphase step that occurs in meiosis 1. Mitosis, you only have 1 round, so whenever we're talking about metaphase and mitosis, you're not going to see a Roman numeral 1 or 2. All right, and so for this one, I would definitely learn this slide, understand this slide, because it is really helpful to understand the differences between mitosis and meiosis. And there's another one like this highlighting how genetic diversity is created in meiosis. Okay, so here the main thing is the metaphase alignment. So in mitosis, replicated chromosomes line up. And replicated chromosomes are the little X's, right? Here they're not so much X's, they're like 2 chromosomes that are just next to each other. But the X's and these little chromosome sandwiches, what they represent is 2 replicated chromosomes joined at the center by a kinetochore, which you don't have to know. But in mitosis, you get replicated chromosomes lining up. In meiosis, you get replicated homologous chromosome pairs lining up. So here you have chromosome 1 from mom and chromosome dad from 2. They've been replicated and they pair up and they align in the center, right? And you can see that in the diagram. You definitely look at the diagram to understand what I'm saying, or Google replicated homologous chromosome pairs, metaphase in meiosis, or something like that. But hopefully that's clear. All right, what gametes are produced in spermatogenesis and oogenesis? This is actually from, this is not from chapter 6, this is from, I think, lecture number 12, or lecture number 11 on human reproduction and development. And basically, sperm gametes are generated via spermatogenesis, and egg gametes are generated via oogenesis. All right, crossing over and random alignment. This stuff is a little tricky, but it's really helpful to look at this slide and understand the difference, or not the differences, but how meiosis can generate genetic diversity by crossing over an independent assortment. It kind of illustrates it for you, right? So spend time on this, looking at crossing over and independent assortment. But that first question was, what 2 processes during meiosis 1 contribute to genetic diversity? And the 2 major things in meiosis that contribute to genetic diversity are crossing over and independent assortments. So another word for independent assortment is random alignment, which I think is what was listed there in the review. Okay, then the next one is, what gets shuffled during crossing over? We'll answer all 3 of these questions in the next slide. Between what kind of chromosomes does crossing over occur, and at what step of meiosis 1 does crossing over occur? So we'll kind of answer them in reverse order. So crossing over occurs in prophase 1, right? There's a little picture from your textbook with the little hand. That's the picture of prophase 1, where these replicated homologous chromosomes condense and pair up. Then if you look at the figure on the bottom, the crossing over is occurring between replicated homologous chromosomes, right? Between that replicated homologous chromosome pair. So those are the chromosomes that crossing over is happening in. And then what does it shuffle? It shuffles, crossing over is like shuffling the alleles between homologous chromosomes. So you're shuffling the uppercase alleles and the lowercase alleles between the blue chromosome and the red chromosome, right? And in this example, I think all along we've been saying the blue chromosome is from your dad and the red chromosome is from your mom. And so the alleles in those get shuffled. And the important thing here is that this contributes to genetic diversity. And the other way meiosis contributes to genetic diversity or generates genetic diversity is independent assortment. And here's the slide. Really, I just bolded the part that I want you to focus on. So independent assortment is when homologous pairs align in different arrangements. So you have the arrangement, one possible arrangement on the top of the figure and another possible arrangement on the bottom. So you can see that you simply reverse the order of that second set of chromosomes, say they're chromosome 2, where instead of having red on top and blue on bottom, you have the opposite, the reverse. And if you look at the gametes that results after meiosis, you can see that with this second arrangement of how these chromosomes are aligning, you are now generating another combination of chromosomes for these gametes. So again, depending on how you arrange the chromosome pairs during metaphase, this is happening during metaphase 1, you can end up with lots of different combinations of chromosomes 1 through 23. All right, now chapter 7. Genetic mutation creates genetic diversity. Remember, mutation is the main mechanism by which genetic diversity is created. Then you have meiosis using crossing over and independent assortment, and then finally you have sexual reproduction where you bring in one set of chromosomes from one parent and another set from another parent. And so now you have many combinations. Okay, so in this part, this question here, how do genes fit in the central dogma meal analogy? That actually was listed in chapter 6, but we're going to look at it here. And I'm not going to put a slide up, but you should know the central dogma meal analogy from last unit, how you go from DNA to RNA to protein, and that's analogous to making a copy of a recipe from a master cookbook onto an index card. That would be the RNA, and then taking that index card into the kitchen to make the meal, which would be like the protein. So genes are the instructions for one protein, right? One gene codes or carries the instructions for one protein. And then what is an allele? An allele is, and how are alleles generated? We'll answer those two questions in these next few slides. But here I highlighted when you fertilize egg and sperm and you generate a zygote, this new zygote may have two different alleles for each gene, right? Because you have chromosomes 1 through 23 from the mother and the egg, and chromosomes 1 through 23 in the sperm, and when they come together, now you have 46 chromosomes, one set from your mom and one set from your dad. So because you have two sets, each gene, you have two alleles for each gene, and they may be the same or they may be different, right? Eye color is always the easiest example. You may get blue-eyed color alleles from both parents, or you may get one blue-eyed color allele from your mom and brown-eyed color allele from your dad. So you may have different alleles for each gene. Now let's look at what alleles are. So alleles, again, are different versions of a gene, and they arise by mutations. So remember, mutations happen in DNA, and that affects the sequence of amino acids that make up a protein, right? Because remember, DNA is transcribed into RNA, and RNA has these little 3-letter codes called codons. And these 3-letter codes, it's a 3-letter sequence of nucleotides that specifies an amino acid. So if you introduce a mutation in those 3 letters, say you switch one of those, that might now specify a different amino acid than what you originally had. So on the bottom here, there are 3 examples or analogies for mutations. And the first one is a mutation that doesn't affect the protein sequence. So here you make a change in the DNA sequence, but that doesn't affect the amino acid sequence, right? There's some mutations that will not affect the codon. They'll change a letter, but it still will specify for the correct amino acid. The second example is a mutation in the DNA sequence that does affect the amino acid. But it's still, the protein is, even though it has a different amino acid than what it normally had, it's still functional. And then the third example is a mutation in the DNA sequence that changes the amino acid sequence, but also changes the function of the protein, where maybe it even destroys the function of the protein, right? Now, a good example of alleles, and so these would all be alleles. Let me backtrack a second. All of the changed spelling of the words there on the bottom, the mutant allele, right? The After the mutation is introduced, those are alleles. Those are different versions of that gene, and they can be functional, non-functional, but they will always be different because they have one different letter in there, one mutation. So a good example of this is eye color. It's thought, scientists think that all eye color used to be, or the original eye color gene used to be for brown eyes. And somewhere along the line, that brown eye color gene acquired a mutation that destroyed the function of that eye color gene. And so then eyes were colorless and they became more bluish. So you get blue eye color and then other versions of that green eye color. But those blue eye color, green eye color genes, those are alleles of the eye color gene. Because remember, an allele is just a different version of a gene. So you have brown eye color gene, blue eye color gene, green eye color gene. You don't, an individual can have only two alleles, right? One from mom, one from dad. But a gene can have multiple alleles. So eye color can have, for example, 5 different colors, but you as an individual will only get 2 of those, 1 or 2 of those. All right, let's move on. So Mendelian genetics, when the role of genes is clear. This is the title in your textbook of this section. Know what genotype and phenotype mean. So here's the slide where we covered this. A genotype represents an individual's 2 alleles for one gene. So in this case, we're talking about pea plants and specifically about seed color. So you can have an allele for yellow seed color or an allele for green seed color. And this genotype confers a phenotype or observable characteristic. So depending on the combination of alleles that you have, that's going to express itself outwardly, visibly in the organism. In this case with seed color, but same with your eye color, right? Depending on the combination of alleles, that's going to express itself visibly in your eye color. So if we look at the chart to the right, in the middle there, you see the heterozygous combination of alleles, where a heterozygous combination is where each allele is different. So here you have uppercase G and lowercase g. And uppercase G is dominant, which is the yellow color. So that genotype, uppercase G, lowercase g, is going to result in a phenotype, which is going to be yellow because uppercase G is dominant. All right, next question. So what determines the traits that one receives from your parents? So this is kind of, this question isn't the clearest. I apologize for that. But what I mean here is the traits here are the outward traits, the phenotype, right? For example, eye color. So what determines the eye color that you receive from your parents is those are the alleles that you get. So that's the simple answer there is alleles, the actual genotype of an organism is what determines the outward traits or the phenotype of the organism. All right, what kind of alleles determine dominant traits? What kind determine recessive traits? It comes from these set of slides. And here, dominant alleles exert their effects whenever they're present. So dominant traits are determined by dominant alleles. And here recessive alleles or recessive traits are determined by recessive alleles. So the example here, the figure on the bottom, you see seed color. So you have a plant that came from a green seed, a plant that came from a yellow seed. You pollinate the plant that came from the yellow seed with pollen from the other plant, and you get all yellow seeds. So here you can see that the yellow seed color is dominant. It dominates or overpowers the other allele, the green seed color. So because yellow is dominant, that must mean that green is recessive because it is overpowered by the yellow color allele. All right, last question. Why do identical twins share 100% of the same genes? And we covered this in the last slide of lecture 12. And on the right, you see identical twins, how monozygotic or identical twins come about. And as the name monozygotic means, it means that they come from one zygote. Mono is one, zygotic is talking about zygote. So here you have one egg and one sperm that fertilize to create one zygote. That zygote develops into an embryo, and that embryo somewhere along the line is going to divide into two embryos. So now these two embryos share 100% of the genetic content, right? They are genetically identical. And so as they continue to develop into fetuses and babies, these are going to be identical twins. They're going to share 100% of the same genetic content. And if you contrast that with fraternal twins or dizygotic twins, these are twins that come from two different eggs that are fertilized by two different sperm. So they're basically as if you and your sibling all of a sudden were in your mother's womb at the same time, right? But that's the difference between fraternal and identical twins. Okay, so hopefully that helped review some things that might not have been clear. Again, for the next review, I encourage you, if you are going over that review sheet and you realize that there's a lot of stuff on there you're not too sure about, please email me some questions and ask me about it. All right, hope you guys have a great weekend studying. Study hard for sure, and email me if you have any questions, and I'll do my best to answer them. All right, peace and grace. Bye.